Welcome to Quantum Photonics. The convergence of AI, recent advancements in biology and stem cell technologies have opened up new horizons in the field of synthetic biology and wetware computing. Finalspark, a pioneering Swiss biocomputing startup, has launched the first ever online platform enabling global researchers to conduct experiments remotely on biological neurons in vitro. You will see here at the link that I had posted on the room chat that you will be seeing the live um, link to the biological neurons in vitro in Finalspark. So please check that out because that is very, very interesting. So the platform provides around the clock access to 16 human brain organoids aimed at developing the world's first living processor. These bioprocessors, composed of living neurons capable of learning and processing information, consume a million times less power than traditional digital processors, potentially reducing the environmental impact associated with the rising use of computers. So, with us here today is Dr. Fred Jordan. He is an inventor, entrepreneur, and a scientist who holds a PhD in signal processing from EPFL in Switzerland. He is also the co-founder of this company, Finalspark, as well as the CEO. So, Dr. Jordan is specializing in the image processing technologies, and he is also co-author of 18 peer-reviewed publications in over 80 patents. His first company, Vision, was a great commercial success in digital authentication technologies, and in 2014, he co-founded Finalspark together with Dr. Martin Cutter, and over his career, he developed a deep expertise in digital technologies and synthetic biology. So, yeah, with us here to tell us about their very interesting work is Dr. Fred Jordan, whom I would like to welcome in Quantum Photonics. So, Fred, welcome to Quantum Photonics.
Well, thanks for welcoming me here. It's the first time in Clubhouse, so I'm discovering this way of interaction.
Yeah, thank you for coming here as well. And yeah, this is the first time that Dr. Jordan came to Clubhouse, and I hope that this will be a pleasant experience for him. So, Fred, we have put the slides up at the top of the screen. So, yeah, I'd like to pass back the mic to you. We are very eager to hear your presentation.
Yeah, thank you. Just trying to see if I can see the slides. Yeah. Okay. So I guess I will go through the slides. And I will give the number of the slides so people can follow. I guess you're able to see the slides now.
Each of us has individually downloaded it, so if that has happened, yes, the slides are beautiful.
Well, thank you. So I will assume that people can watch the slide as I go through them. It's a correct assumption.
Yes, indeed,
Yes.
sir.
Yeah. Okay. So thanks. Yes. So the purpose of what we are doing is to build the first bioprocessor. Okay. So well, on slide one, there is the team. So it's a small team. Okay, you have to understand that this is a startup. So we are 6 people, and we are based in Switzerland, and we've been working in this field of research since 2014. So we switched to the field of biocomputing in 2018. And the reason why we did that is that we realized that there was way more potential for biocomputing than with artificial neural networks. Um, okay. So on slide, the next slide, on slide 2, you can see, yes, the goal that we have here, it's really to move from digital processor to bioprocessor. So it's a radical move, and we don't actually intend that bioprocessor could replace digital processors for every task. I don't believe that you can run Windows 10, for instance, or iOS on a bioprocessor. That would not be adequate because of the latency, which is typical of living neural networks. So what we are trying to do is really to use human living neurons as a way to process information when they are connected in a network. And this kind of network, there are different ways to do it, but we are working specifically on so-called brain organoids, which is a three-dimensional arrangement of those neurons. And on slide 3, these are the basic reasons. Why are we using actually living neurons? The first obvious argument is that, well, they work quite well. You know, if you look at our brain, it's an impressive demonstration that processing of information through living neurons is actually effective. Okay, this might sound as trivial. Consider the alternate proposition, which is to make a cognitive system based of artificial neurons or based on quantum computers. These proposals are way more challenging, actually. Somehow, using living neurons is a very conservative approach. Okay, and as mentioned, also there is the interest regarding energy consumption. If you look at our brain, it's typically a consumption of about 20 watts. But if you wanted to make a digital simulation of a brain, you would look at a small nuclear plant, actually. This assuming that we know exactly what to simulate, which is not even the case. So in this world, this is typically very interesting, very appealing proposal to have something that can do processing for 1 million times, processing of information for 1 million times less energy, obviously. Okay. And there is the third argument, which is the scalability. When we use neurons in our lab, we have actually to stop the multiplication of the neurons, not in order not to have too many of them. These are von Neumann machine, you know, they replicate infinitely, I would say. Well, virtually infinitely, I would say. Okay, so that's also a very compelling argument in favor of using living neurons. On number 4, I just show what kind of vision we can have is the biocomputing in the cloud. So I don't foresee short term that you could put a brain organoid in your smartphone, for instance. That may become true maybe sometimes in the future. But if we want to be conservative, what we have already built with the neural platform is an example of remotely accessing to processing using living neurons. And this is quite realistic to do because you centralize all the resources to keep our neurons happy. And I can tell you this is not an easy task. On the slide 5, I really show what we're talking about. Okay, we have on the left hand side, you can see electronic microscope picture of the brain organoid we are typically using. So it's about, I would say, 400 micrometer of diameter, and it's spherical. And in there, you have, I would say, in artificial neural networks terms, you have the connectivity and you have the neurons. On the central part, you can see actually what it looks like when you get a zoom in of the complexity of the network we create. So understand that when we create those brain organoids, we have absolutely no control over the connectivity between the neurons. So this will happen later during the so-called training stage. So on the right picture, it's an immunofluorescence picture, which is interesting because it gives us some insight of what is inside the brain organoids. So this is called forebrain organoids because it's this type of neurons that we find in the forebrain of human brains, actually, because these are human neurons. So you can see in red the mature neurons, and in blue you have the astrocytes. So just as a reminder, you have neurons and you have glial cells. So if you look at the microscope, it's not that obvious to make a difference between a neuron and a glial cell. But there is a striking difference, is that the neurons can spike, that means they can generate action potential, and the glial cells cannot. So for instance, two types of glial cells are the astrocytes and the oligodendrocytes, for instance, which commonly are considered as helping the neurons, but this is very debated in the sense that for many years, neurons have been considered as I would say, the stars of the brain. But it may be that a number of processing is also happening in the glial cells. So again, our conservative approach is to use all of them and to have something that mimics what we have in our brain in order to ultimately get a similar processing power. So the slide 6 for me is very interesting in the sense that it's really how it looks in real, in visible light. Okay, so you have, so what you see in the background are the electrodes, the conductors of the electrodes. Then you have a circular transparent shape, which is the membrane, on which is deposited the brain organoid. So you have to imagine actually that there is a hole in the center of the membrane that enables the brain organoid to be in contact with the electrodes. And on the slide 7, I show this in more details. So you can find all this in way more details in the publication we did in Frontiers, which was also given as a reference to this discussion. So what is interesting in the particular setup that we are using, it's called the air-liquid interface. So you have the medium. So the medium, it's basically water, okay, with amino acids, vitamins, salts, glucoses, which is required to keep the homeostasis. That means to keep the neurons living, actually. So above the medium, you have membrane, which is a porous membrane. And then the medium can go through this porous membrane And then you have electrodes. And finally, you have what is called here in this slide the neurosphere, which is the brain organoids, let's say. And by capillarity, you have actually a very thin layer of medium that covers also the brain organoid. And above this is air. This is why it's called air-liquid interface. So it's a very particular setup, okay, that we have here, and it's very efficient because we just have to deposit brain organoids, and in 15 minutes we can make some experiments. On slide 8, so Sylvia also gave the website address. So if you go on our website, in the live section, you will see live images and signals coming from our lab, which is just below where I'm talking, actually, in Switzerland. Okay, and so this picture is changed every hour, and you can see here on this part, this is called a multi-electrode array, MEA, and you have actually 4 circles which represent the membrane, and in the center of the membrane you have a hole, and you see a sort of shadow inside the hole, and this is actually where the brain organoid is sitting. And you have 8 electrodes coming to each brain organoid. So you could see this as a, I know, a 4-core processor, if you want. So on the slide 9, you see actually the whole setup in order to keep them living. So you have the organoids with the electrodes. This is actually sitting in an incubator, which guarantees that we have a constant temperature of 37 degrees, 80% humidity, and 5% CO2. The CO2, which is 100 times actually more than in our atmosphere, is there to guarantee that we keep the pH of the medium neutral. That means around 7. So you have these famous electrodes, which is the main way of interacting with the organoid. And of course, from the electrodes, we have converters, analog to digital, and going to the computer. And but back, we have also digital to analog converters, and these are used to stimulate the neurons. Okay, so you can actually read signals, which are typically in the range of 50 to 100 microvolts, I would say, and send stimulations, which is typically in current stimulation around a few microamperes. So it's very, very low. Okay, so and in between, we are going to make some processing to know exactly which signal to send at which time on which electrode in order to change the behavior of the organoid. So and maybe I can make just a pause here. I just to mention that the purpose of all what we're doing is to train the neurons to do something useful, but it should be very clear that it's not something that is working now. That means you cannot program easily those biological neuron networks. It's still a topic of research. So this has to be clear because you can find a lot of things on the internet like if it was working right now, but it's not the case. It's a topic of research. So this has to be clear. So well, on the slide 10, we see actually basically the complexity of the microfluidic system that we had to develop. In this microfluidic system lies a bit of the secrets that made up the possibility to have these neurons living for several months. This is what we have now. At the beginning, it was like 1 hour, 1 hour and they're dead. Okay, so now we have sort of autonomous system, and the less you touch those neurons, the better it is. And there is we learned a lot of things to have those neurons happy. And one of the critical things is to have a constant flow of medium. You remember the medium is going below the organoid, and it's now flowing 24/7, like a few microliter per minute. So it's very slow, but it never stops. Okay. And we have also waste collection and all the system that guarantees that automatically we try to simulate artificially an homeostatic system. This is what we are doing here, actually here. So this is number 10. So on number 11, it's just a snapshot of what you can see in live, actually, on the website of Finalspark. We see the signal. So just that I explained on this slide 11, on the top left, you can see, for instance, 8 graph. And this 8 graph corresponds to the 8 electrodes of one specific organoid. And each graph is a record of 1 second. Therefore, all the graphs are updated every second. And this is 24/7 on our website. You can see what's going on. Okay. So if you look on electrode 23, for instance, on the top left, the top right, sorry, you can see that electrode 23, we have a train of spikes. This is spontaneous activity of the neurons. Okay. So there are 2 types of activity. So when we talk about activity, we are talking about the change in the electrical field that we are measuring following an action potential. So when an action potential is created by one neuron, it changes the electrical field that we can measure with the electrodes. This is what we are measuring here. So these are spontaneous activity, and the other type is elicited activity. So elicited activity is what you get when you send a stimulation on one electrode, and maybe some electrodes are going to reply to this stimulation. So, and they will not always reply the same way, by the way. There is a lot of things in the state of the art about this. So on the slide 12, so it goes more about how to do research with our platform. So just putting here a very simplistic diagram that we are actually using sometimes, which would be how to train the neurons in order to spike more. Okay, I'm not saying in any way that this thing that I'm showing is working, and in the sense that it always successfully trains neurons. Okay, it may work sometimes, sometimes not. So again, still a topic of research. Okay, I'm going to do a lot of understatements here, very, very conservative statement of what is possible today. So here, before I explain the diagram, I have to mention something else. There is a third way that we have for interaction with neurons, which are neuromodulators. We are using specifically dopamine. Dopamine is well known in the state of the art as critical neuromodulators for learning in vivo. Okay. Remember, in case you don't know, that there is almost no state of the art regarding learning in vitro. Okay, so as incredible as it seems, if you think about the tens of thousands of papers in neuroscience every year, learning in vitro is, let's say, a new field. Let's say this way. Okay. So here, based on the state of the art of in vivo and the importance of dopamine, we thought that it was critical that we are able also to use dopamine in vitro. This is what we are doing. So just a technical note, how we are doing this is that we put so-called caged dopamine. So caged dopamine basically is a dopamine which is embedded in sort of molecular cage, and we are able to open this cage using UV radiation. Okay, so UV light. Okay, So with a very short spike, like 50 milliseconds of UV, we're able to open this cage and have dopamine released to the organoid. So why is it important? It's important because we believe that dopamine may be, very cautious here, may be one way to provide reward to a system. And one assumption which is done is that rewarding a system may lead the system, the biological networks, to change the synaptic connections in order to reproduce what has been rewarded, the behavior which has been rewarded. Okay. So knowing, and the last piece of information is about timing. If you reward a system, a biological system, we also, based on the state of the art in vivo, assume that in vitro, the timing is extremely critical. Therefore, with this type of uncaging system, we're able to be extremely precise on the exact time when we reward the system. Okay. So let's consider now this diagram. Okay. So here we start by send steams to each electrode. Okay. That means we are sending a small current to each electrode. Now we are going to elicit some spikes. Okay. So, for instance, for the next 600 milliseconds, there may be some, on some electrodes, some elicited spikes, some action potentials which are produced by this initial stimulation. We count them. Okay. And the whole thing that we are training the system to do here is to have more spikes when we stimulate it. So we have a condition, if n greater than n prev, then we are going, if n is greater, we are going to reward the system. In order to reward it, we are going to switch to UV. Okay, and the idea is that we inform the biological network that it has been doing great here, and we reward it. Okay, it's very layman terms, but this is actually what we are doing. Okay. Then there is some purping and things occurring because this way to simulate a little bit the reuptake which naturally occurs in living system when dopamine is released. And so on the left hand side you have exactly the equivalent in Python. Okay, and the reason I put this in Python is that Python is at the core of the research platform that we make available for researchers. What researchers are doing is that they are writing Python script, where they develop some algorithms to try to find the optimal combination of stimulation and analysis of signals to have exactly the reply they want. So on slide 13, we have actually just a screenshot of this publication in Frontiers that goes in way more details about what we've been doing there. On slide 14, just to give you some sense of who we are and the lab that we are using. This is a classical cell culture lab. Okay, someone who doesn't know anything about biocomputing but only knows about cell culture would find that it's absolutely standard environment. What is surprising are the pumping system and all the electronics, which is sitting also somewhere here. So this research platform for universities who work on the topic of biocomputing, of learning and training those neurons, the access is free. Okay, so so this is the list of universities on slide 15. So 9 universities right now are using this platform, and from time to time we are even giving courses to teach to people how to use this platform. Just to be clear, this was not the original purpose of Finalspark, actually, to share this platform with others. It's just that during the COVID, we didn't have a possibility to come to the lab, and because of this, we developed this system to work remotely from home on the neurons. And at the end of the COVID, there were like 34 universities who showed some interest to use our platform. And so not only for us from home, but really from all over the world. Then we realized that given the complexity of what we try to achieve here, it would be a nice idea to have an open innovation approach. And this is how it started. Okay. Well, slide 16, just to show that what is interesting is that this field, I would say, is new. The idea itself is not new. You will actually find this in science fiction movies from since a very long time. There's been some isolated work, and isolated is a keyword here in the past 20 years of people looking at this. But there was actually no publication or no collection of publication that was specifically dedicated to organoid intelligence or wetware computing. Okay, and Frontis created the first collection last summer of last year, and this was the first ever collection. So it's that new that it was not recognized as a field of research until we'll say 1 year ago. So that's really astounding. But there was also a very important publication last year by Professor Hartung in the Financial Times. Well, that was taken by the Financial Times. That showed the potential financial interest also of moving computations to biology for some types of computation. Okay, and basically that ends what the presentation I wanted to do.
Yeah, thank you so much, Fred. Amazing slides and great explainer. So, we'd like to announce to the audience that we are opening the floor for questions. If you have questions, please feel free to raise your hand. We will bring you up the stage. And we would like to welcome those who raise their hands. We have Ihab and P here on stage. Yeah, but I'd like to ask one question to our speaker before I pass the mic to them. Yeah, Fred, I would just like to know, are you scaling this up soon, or what is your next step um, on this project that you have?
So by scaling up, do you mean the scale of the organoids or the number of users?
I think both, on both sides. I'm just curious.
So for the organoids, we are not ready to scale up because we have well, there is very well known vascularization problem when you create organoids. So the field of organoid exists, independently of us. Okay, it's a big field of research, and the people have this problem that you are unable to get nutrients inside the organoid. So if you are more than, let's say, 1,000 micrometer, say 1 millimeter, you start to have necrosis at the center. So this is a well-known problem that we are going to tackle for sure. But at this point, we are not on this yet. Okay. Now for the other type of scalability, which is the number of users, what happened in the past 2 weeks is that some companies started to ask for paying a monthly fee to access to our neurons and to be independent. So they want to do their thing and they are ready to pay for this. Also, this was not part of the plan. So we are actually scaling up. So just last week's we welcomed actually our first customer. There will be another one that comes, I guess, in the following 2 weeks. But we want also to keep all the universities. So we are rapidly scaling up on the electronic side. And I guess at the end of the summer, we should have 6 multi-electrode array in parallel, each with 4 organoids. And we've been working actually for a few months because we were anticipating that that this may go well. So we've been working a lot on the electronic side to be able to have many users at the same time. So I guess we should be able to welcome more and more users on this platform.
Yeah, thank you so much. If there is time later, I would like to ask more questions. This is very interesting indeed. And for the sake of those who just came in, I would like to reintroduce our guest speaker, Dr. Fred Jordan from Finalspark. He is an inventor, entrepreneur, and a scientist. He holds a PhD in signal processing from EPFL in Switzerland, where he specialized in image processing technologies. He is also co-author of 18 peer-reviewed publications and over 80 patents. His first company, Altvision, was a great commercial success in digital authentication technologies. And in 2014, he co-founded Finalspark together with Dr. Martin Cutter. Over his career, he developed a deep expertise in digital technologies and synthetic biology. So, we'd like to pass the mic now to Ihab, who came in. We are doing giving the opportunity to speak in PTR order. So, yeah, Ihab, mic to you.
Okay, thank you. Amazing stuff. Signal processing and digital image processing. Amazing work. I don't have much chance to see what's going on. Multitasking here at work. So, Professor Fred, if your image processing, do you use like Fourier curves? I like the mathematical algorithm. Like if you change number values to numerical values. If you can tell me what do you do? Thank you.
So you mean processing digital information with the neurons?
Yes.
Yeah. So the processing is very elementary. You know, first thing, there are two categories of work being done in biocomputing. Because I make this thing just to answer your question. I don't know if you know reservoir computing or liquid state machine. This is a way of processing information by using the natural capabilities of a system. Okay, I give you an example: water. Okay, you can process information with water. Okay, I explain this. There is a wonderful publication which has been done many years ago with speech recognition in a bucket. Okay, where people use actually a microphone to drive some oscillators on the surface of water, and then they have a camera when they take pictures of the waves that the water creates. Okay. And then using these waves, they recognize the voice. So instead of processing the 1D signal, they process 2D signal. Okay, and this processing for 1D to 2D is, I would say, virtually for free. It's done by the characteristics of the system, and this is an example of reservoir computing or liquid state machine. So the reason of my introduction is to answer your question. So there are a number of people who are actually using the neural networks, living neural networks, as they are to process information. Okay, that means the system is like it is. Okay, and you can use it like, for instance, if you notice that when you send a signals, you get on one electrode, you get two signals on another electrode. This is already a processing which is done. Okay, and you can use it. Okay, so it's one way of processing information is to use the system as it is. Okay, this is not what we are doing, but if you look in the literature, the very limited literature of people working on wetware, you will see some people doing this. Okay, what we are trying to do is way more challenging. Actually, is to change the behavior. So if I want to spike on an electrode when I spike on another one, and I don't have what I have, the challenge that we put on ourselves is to change the behavior of the system. And so we So in this sense, sometimes we succeed and we are able to do the kind of digital processing that you mentioned. Most of the time we fail. So so this is why it's a topic of research.
Okay, great. Just a follow-up question. Are you familiar with the EEGs, level of sleep analysis, alpha, delta, and key spindles?
Not very much. Not.
Okay, because I think like my background, I'm a gastroenterologist, and I'm into medical AI right now as we speak. I think us as a humanity, we need more about sleep analysis and biofeedback with EEG, electroencephalogram, and it's pretty much you work with neurology or neurons, and pretty much that's your alley. So your feedback, please, if we can think together about this. Thank you,
Yeah. So we've been contemplating the use of EEG in our case for some time, indeed. We have also, if you talk about sleep, we have been also contemplating circadian cycles in our case.
Circadian rhythm, and you're gonna use hormonal parameter markers, or you're gonna use just timing.
Where we would like actually to induce circadian cycles rhythms. We believe that it may be necessary to have this kind of cycle, to have organoids working well in the long run. Okay. So this is a topic of discussion. It's at Finalspark for sure. For EEG, we have not looked at the device yet. Also, please also take into consideration the number of neurons that we have. Okay, because when we have EEG, I don't know how many neurons are contributing to the EEG, but in our case, we are about 5,000 to 10,000 neurons in our system. Okay, so it's a very low number of neurons.
That's 4 times what Neuralink. Neuralink have 1,600 to 1,500 channels, so that's still a good number.
Yeah, so this is just the number of neurons. Okay, yeah. So yeah. We actually monitor through 8 electrodes. This so we have a restriction here. So
thank you, amazing work, and that's exactly. If I have a time machine, I would always go to that. Like I love signal processing. I always want to be an electronics engineer. But my dad, God bless him, I'm gonna be an MD. Okay, fine. So here we are.
It's very good. Thanks for the nice words.
Absolutely. Thank you. Thank you for everything. Great day.
Yeah, thank you, Ihab. And we'd like to pass the mic to B.
Thank you so much, Fred. It's an honor to share the stage with you. And Cecile, thank you for your continued just great guest. I really love quantum photonics. Thank you so much. Fred, my question, I first learned about your company a couple of weeks ago. There was a viral tweet that was talking about your platform. It made the claim that, that Finalspark organoids use 1 million times less power than a digital processor. So wondering how accurate that is, but also just wanted to reflect a little bit on, the potential energy crisis that the increased use of AI may be causing. And we hear about, all these nuclear power plants possibly being built to accommodate the increased demand for AI energy. And if we had, biocomputers that, required 1 million times less power, obviously that would be a major potential solution, and nuclear power plants take a long time to come online, etc. So I was just wondering how, I know we were talking a little bit about scalability and the manufacturing process of the organoids. Just wondering, like, if you've had any conversations that, where there are projections about how much of the AI energy portfolio problem, what your platform could do to help with that problem, And how much do you think you could contribute, say, in the next 5 to 10 years with that?
Okay, thanks for the question. It's a very challenging question. There is at least a part of your question which I can answer is quite easily. Is the 1 million number was initially with I mean we have to be very cautious about this time this kind of numbers. Okay, but journalists love them. But it was initially stated in the Artung publication that I was referring before about organoid intelligence when comparing the equivalent consumption of digital versus biological systems. And you're right that these biocomputers, well, I would say virtual biocomputers, okay, come exactly at the right time. Okay, so that's very good news. I fully agree with this. You tell me, you asked me which fraction actually of this this power consumption we could handle, we could transfer to biocomputers, and this I really don't know. What I can say is what is not going to be possible. Okay, so I was talking about latency before. Okay, latency is going to be always horribly slow on biological systems, and I reasonably suspect that arithmetic computation is not going to be very good for biological neuron systems. Okay. Now, what we have to consider is that with the increase of AI actually is using simulations of neurons. Okay, so we are actually running computation in order to simulate neurons, and because there are many neurons, this consumes a lot of money and a lot of energy. And if you think about it, it's quite absurd in some sense. If you have the real thing, that means the real neuron, which is available and that you can use it, and that consumes, let's say, 1 million times less, I mean, it's a very compelling proposition to say to use the real thing. Okay, so because we use many multiplications and addition just to simulate those biological neurons, and I must also add two things. Learning. The learning of artificial neurons is extremely intensive from an energy perspective. Comparatively, the learning of biological systems is extremely conservative. You can see this also by the time it takes. I mean, the analogy is not very scientific, but just let's share intuitively. For a child, you show to a child 5 images of a dog and 5 images of a cat. He knows basically what is a dog and a cat. Okay, it's not going to run with these numbers with an artificial deep learning or convolutional network. You have to show like 5,000 of each, and one small details can make a whole of difference for an artificial system, because he has no fundamental approach of what he's looking at. So in this sense, there are a number indeed of use of AI today which could be translated to biocomputers, but which fraction, I'm sorry, I don't know. But I must say something else. If you had talked with the inventor of the transistor, like Shockley, what would be the inventions? Okay, beginning of last century. I don't think he would have thought about smartphones and the internet. So actually, maybe the most striking applications of wetware computers are not the ones that we can imagine today.
Thank you so much.
You're welcome.
Thank you, B. And I think there's someone who had a comment and has a question here at the room chat. I think Xiao Chun. I will read your question in a while. You are also welcome to come up the stage. So just in case you want to go up the stage, please raise your hand. Otherwise, we will just read your question. So I'd like to give the mic to Kevin now. And yeah, thank you for coming up, Xiao Chun. I'll mic to you after Kevin. So Kevin, mic to you now.
Oh, thanks, bonjour. This is quite amazing, and it's like the biggest, so groundbreaking. I'd like to I did some research and tell me if some of this is correct, and if you can maybe speak on it. Saw that your neural platform uses an architecture that can be classified as wetware. The mixing of hardware, software, and biology, and the main innovation delivered by neural platform is through the use of four multi-electrode arrays, MEAs, housing the living tissue organoids, which are 3D cell masses of brain tissue. And these organoids can perform simple computational operations and store then recall memory. In principle, they provide the sort of control we need to design and build real cellular computers. So I don't want to go on forever, but and then the operation of these bioprocessors is not limited to just computing. They can also detect changes in biological systems and report their findings back in accurate and reliable analytical information. And this makes them incredibly versatile and capable of being used in a wide range of applications, from smart materials to new kinds of artificial intelligence. I want to see if I'm right about all this. And like biopharmaceuticals, specialty products and industrial chemicals, environmental management aids, biofuels, and bio remediation, food and beverage industry, goes on and on, pharmaceutical, of course, bioinformatics, and nanotechnology. And so I don't know if you can speak on if I'm correct with all that. It just blows me away.
Yes, first I must say thank you, and it blows me away also. Okay, and I really feel like I'm living in a science fiction movie every day. About the application, there is another one which is also interested. It is out of our initial target, but I just mentioned it. Okay, consider that we are using the neurons that we grow through stem cells of a donor. That means that we can grow the neurons of everyone, and we can make a small version of some parts of its brain. That means it's possible with this system to check the effect of a drug. For instance, if you have a disease, it's possible to check if it helps you, if you have some learning problems, what helps you, what also are the side effects of a drug on different parts of the brain. And this is individual medicine, okay, that will be targeted exactly for your neurons to as a human being. So this is another perspective which can be added here. So I want to mention that we don't take really the direction of pure computing. It's although it's called biocomputing, we are more like if you know about artificial neural networks, at the end it's about input and output, and you want to have the output matching the test samples that you have, and it's exactly what we do. Okay, we have input, that means electrical signals. We have output, that means the electrical signals coming from the electrodes, and if they are matching what we want, that means the loss, that means the difference between what we want and what we have is close to zero, and then we can reward the system. Okay. And it's interesting because, said in those words, there are no fundamental difference between what we want to achieve from an artificial neural network and a biological neural network. Okay. The main difference is not what we want to achieve, is how you change the weight of the connections. In artificial neural networks, you just change the numbers in tensors. I mean, it's not easy, but in our case, we cannot change. So the equivalent of the weight in our case are synaptic connections that we cannot address one by one. It would be unrealistic. You have to reward the system and hope, and this is a critical word, that it will update automatically all the synaptic weight in order to get closer to your target at the next time. So you listed the number of applications. I agree with you. And just wanted to highlight that there is a fundamental difference with also artificial systems when we talk about training.
Oh, thanks a lot.
Thank you, Kevin. Or Fred, I would like to comment. There might be a use case that we haven't even just imagined as of now. So it will emerge in the future, just like when there was the printing press that created a surge of demands in spectacles, and so it led us to create lenses up to the microscopic level, and even led us to do these metamaterials at the present day. So sometimes when you do a research or you have an invention, you do not even imagine what the use case is. But maybe in a few years, we will be surprised that your project has led to use cases that we haven't even thought of.
Yes.
So,
I agree with yes, with this. I think the only idea which is a little bit new, okay, is just to consider that the neuron is a machine that we can use. All the rest is engineering, and that's interesting. Consider when planes were invented, the only idea was maybe we can make a flying machine which is heavier than air. Okay, and then people came with solutions. And what is interesting is the change of the perspective on the systems that we are using, considering that neurons is a small machine and that can be used, gives a new set of applications, of potential applications.
yeah, yes, thanks. So we have here Xiao Chun on stage. So Xiao Chun, I'd like to give the mic to you now.
Hi, Cecile. Hi, Fred. Hi, everyone in the room. I really appreciate your work, Fred. And I'm sorry, I'm really nervous on the stage. I went through all the slides along with your presentation, and I just checked your LinkedIn profile, CDO at Vision. Sorry, L Vision. Exactly as Brian has mentioned. And I hope that your business will be successful. Just now, I put my comments in the chat because I'm really afraid that I will ask a silly question. During your presentation and your conversation with Kevin, you mentioned about the reward system, and I was really curious about in the dopamine example, I'm afraid that I didn't really capture your explanation about this part. And how can we confirm could we confirm the reward system is built by the activation of dopamine circuit? If it is in this case, how will the system regard this circuit as a reward? Because there's no a sense such as the sense that will be generated in a human being. So yeah, that is my question, and thank you. Sorry, I'm really nervous.
So first, this is by far not a silly question. It's really a point-on question. This is what you ask is really the critical question. Okay, how can we assume that dopamine is going to be perceived as a reward? And even though, even if it's perceived as the reward, how can we assume that we are going to have an update of the synaptic connections in order to reinforce the behavior which has been rewarded? Okay, and I'm sorry, I cannot give you direct, statement on this because the science on this, that would be in vitro science of learning. I mean, put very briefly, does not exist. The science in vivo is huge. I mean, it's 50 years of state of the art. It's wonderful. But we've not been able to find answers to exactly the question you asked. And at this point, I'm afraid we have no other possibility than to try and characterize the results. And this is already complicated. Just to give you an example, there are, as you certainly know from your question, there are 5 dopamine receptors. Okay, and we have seen that in some of our brain organoids, we have only the D2 like receptors. So maybe that's not enough. We need the D1 like receptors. So we need to do new neurons with D1 like receptors. So the D2 are inhibitor, the D1 are more excitator, but it's not the topic. The topic is more, is it going to be perceived as a reward? Is it going to reinforce the behavior? We don't know. But just to put you in perspective, our direct concern right now is at least to make sure we have the right receptors, so that we have the minimum set of conditions for the neuron to perceive correctly the dopamine release. So sorry if I cannot give more accurate information, accurate answers to your point on question. Very good.
Thank you for the reply. I'm curious about this part because I was thinking that in a simulation of imitation in a machine, how to say, to imitate a brain, we put input output. We make it as an input and output system. So the objective is to modify the behavior, and so that was the reason why I asked this question, because if we have a reward system, so according to the reward, we will modify our behavior. But in this case, not really sure how can we modify the behavior once there's a dopamine present. So that was my question. But I think that probably there is already a solution in that. It's just how to say it. It's quite mixed up. Yeah. I wanted to say that I don't know how can we modify the behavior according to the presence of dopamine. So that was the reason. I don't know whether I well articulated. Oh my god.
Yeah, there is a lot of work which has been written about the reward prediction error, RPE. And that's interesting. So it seems that dopamine is released in a proportion as the difference between what what the system expects to have and what is actually having, and is making a prediction on what is going to be his result, his output, or his of the quality of his match compared to the goal, and this is the brain is known to encode this difference. We thought a lot about this. At the end, we concluded it doesn't help us to know if dopamine is going to drive a synaptic modification in the sense of decreasing the loss. If we talk about with artificial neural networks terms, I don't know how other ways to do than to try.
Go ahead, Sharon.
Thank you for your
Yeah,
Sorry,
thank
go
you so
ahead,
much.
Cecile.
Go ahead.
I want to say that thank you for your reply. I really appreciate your work again. Yeah.
Thank you for your question.
Yes, so thank you. I think there are more questions on the room chat, and I was looking for Jaden if he's still here because earlier in the room chat he had mentioned about the dish brain, and yeah, it is actually from Monash University in Australia, and we had invited Dr. Brett Kagan from Cortical Labs. He talked about this. If you will look at the replays here in Quantum Photonics, they created the first sentient open close quotation lab grown brain in a dish, and they had taught them to play pong. So well, of course the objective of their experiment is different from this experiment that Fred's team has. So, but they also use the neurons as well. I would just like to articulate that. So probably we can ask them again what's new on their project and invite them again. But anyway, this study of Fred has gained, yeah, just like what B said earlier, it had gained a viral status. So that's why I also knew about this project of theirs from publications, and actually it's very popular among the groups that I manage. So I'd like to ask Fred. I think other people have also questions here. Um, I'd like to ask Fred before giving the mic to our other speakers. How is the nutrient and oxygen supply regulated in this bioprocessor environment? And are there specific growth factors or supplements to culture the medium for these organoids? And how do you do or manage waste removal to prevent toxic buildup?
So, okay, so yeah, several questions here. First, very much like the work of Brett Kagan in the company Cortical Lab, and I'm happy to know that we are not the only one working in this field. I tell you, if we were the only one, I would be very, very worried. But I tell you also that there are only 3 companies working on this, and there should be more, I think. So about nutrients, so we are using GDNF, so neurotrophic factors and GDNF, glial neurotrophic factors, at very special stage of the protocol when we differentiate from neuroprogenitors to glial cells and neurons. Okay, normally then we stop afterwards. Regarding oxygen, we don't. Oxygen has been a topic. Okay. Right now we work with the same concentration as in the air. Okay, for oxygen. We have been using nitrogen to artificially decrease the quantity of the percentage of oxygen in the incubator. According to some work, it could help but we didn't see very clear difference, so we came back to normal, percentage. Regarding so the medium that we are using brain freeze and neurobasal plus. I've just said this for the experts. And we move from one medium to the other depending on the. So we have two stages of these brain organoids. Okay, we have one stage when we keep them alive for years, actually, in an orbital shaker. So right now, I think we have about 3,000 of them. Okay, and when we need them, we take them, and we are at this point we are going to change the medium in which they are living to a so-called electrophysiological medium, which is believed, okay, to be more appropriate for electrophysiology operations. So then we have a medium transition when we go from a neurobasal plus to a brain freeze, which is slightly different, with a slightly different osmolarity. And this is the one that we will use then for months once we start experiments with the given brain organoid. Now, waste collection. It's done by gravity, actually. If you look at the microfluidic system that we published, there are actually two microfluidic loops which are interleaved. And one is, instead of using filter, we are just using gravity to have the waste going down in Falcon 50, which is a kind of container. And we let the waste at the bottom of it.
Yeah, thank you. So there is Krish Mazumdar, who has commented here on the room chat. He said bioprocessor is difficult to scale up but can be used for clinical purposes, and how a neuron network or neuroplasticity, etc. works. Personally waiting for liquid neural networks to take some significant step and disrupt all this. So any comment on this, Fred?
You mentioned liquid neural network. Is it? I don't know if it's an analogy to liquid state machine using neural networks.
Yeah, so I'm looking for him. Just in case you're still here in the room, Krish, please raise your hand or make a comment here in the room chat so that we will be clear as to your question. So, in the meantime, I think Ihab had a comment here, and B also said, was it you, Ihab, who asked if they are on the startup process? Yeah, Ihab.
Yes. Hi. Yeah, it's yes. Couple questions. Finally, I got to read your proposal. I'm pulling it up right now. I just emailed it to myself, and I have a quick question on page. God, where did it go? Sorry about that. First thing, are you in a startup phase yet? Or are you round one or two of funding? And how are you guys doing business wise? Money.
We actually, as mentioned at the beginning, actually we created a first company, which is going quite well, and which is called Alvision, and we're using the profit of this first company to fuel Finalspark. Okay, so at this stage we invested about $1.5 million into Finalspark, and there are no other shareholders in the first company, so we are free to use the profit as we want. So that's a blue sky situation, I would say, but that's not enough. We're looking to raise ultimately between 10 and 50 million of dollars. So between 10 and 50 million dollars, it's a lot of money for a deep tech startup, and it will take the time it will take, but this is our ambition. If we don't have this, it's no problem. We'll keep on our current pace, but that's a pity. We think we should have way more people that we can pay to work on this.
Absolutely. So you're about to start round 2. All right, that's awesome. Fundraising. Okay, good news. Are you public? You said like shareholder. Are you public already? Are you like you got the public offering?
No, we are not public. No.
No IPOs. All right. Enough business. I pulled. Thank you. I pulled your article, which is a lot of awesome information. But you have an access to it. So page number 10, research platform for wetware computing. Upper left, fridge for centigrades, fresh medium, sponge pump, rotary valve. Peristaltic pump is like the one in kidney dialysis machine, right? The peristaltic pump. Yes, I do need your help with page 11, reading the numbers next to the signals.
So well, we this is the identifiers of the electrodes. So if you have on the top left you are 8, 9, 10, 11, etc.
Oh,
this is what you are referring to.
Okay. Oh yeah. So this electrode number and this with the signal coming up. Okay. And then I see a part of code like page 12. It looks like you're having logical charts and stems for each electrode approval, and then a code next to it. It's like it doesn't look like a Python. What are you guys writing here?
Yeah, it's indeed absolutely Python code, and it's the implementation of the diagram which is on the right hand side. And the critical part, it says if diff spikes greater than 0, then trigger UV. That means reward. Okay, Mr. Brain organoids, you did right this time. You have elicited more spikes than previously, and we reward you.
Okay, yeah, it's totally my bad because I just pulled it now. I see the electrodes number. Thank you for the explaining. And then slide 16. Biocomputing is becoming hot, and get in touch with us, which I definitely will do. If you can tell me a little bit more about biocomputing, I love to hear more about this. Thank you.
Well, it's so just about the words, first, okay. People are not even sure which word to use, okay. So there is biocomputing, but there is also wetware computing, and there is also organoid intelligence. They are more or less equivalent. By biocomputing, you should know that biocomputing is more generic and includes other ways to compute using biology. For instance, um, uh, um, uh, and RARN, for instance. So genetic codes can also be used for computation.
So can you build me a cyborg?
Sorry?
Just easy. Can you build me a cyborg? Okay, look.
Yeah, but this idea of using biology, well, actually neurons, for computing. I mean, the idea itself is interesting because it's really new. It's the way we look at ourselves which is new, and the consequences of this idea is a number of applications that people that we could not have before. So and I think there are a lot of ways to do this, but I think one of the critical thing at this point is the use of neurons, because intellectually speaking, neurons, actually artificial neurons has led us to believe in processing of information using small entities. And this goes back, actually, now we go back in biological, and we say, yeah, maybe actually we can use this also in biology. But of course we can, because this is how we work. This is not new, but the idea of using it for real is new. And the field of application is, I cannot imagine, actually.
Great, thanks. The very last question, if I may. It's you talk about neurons. Are you working in alpha myelinated C fiber or C fibers, or because I just have to talk about the insulation, the myelin sheaths. It's like you apply electrodes, and which neurons are you using to get your data?
Are you asking which type of neurons we are using?
Yes.
So we are using, well, we're using them different part of the. We try to reproduce neurons of different parts of the brain. Okay. So initially, and most of the publication was done on forebrain organoids. That means neurons which are in our forebrain. We have these past few months accumulated some new results about hippocampal neurons. So from the hippocampus, which is important as far as memory stands. And we will be working in the following weeks on striatum organoids. And just to say the whole story, we also start to work on assembloids. So far, I've been working. I've been talking about organoids. Assembloids is when you assemble different organoids of different parts of the brains. So these are things that we are starting also to do.
Yeah, thank you so much, Fred, and thank you for the question, Ihab. Next time, please tell us more about the assembloids, Fred, when you have started working on it, and yeah, you have made your observations. So we have had been here for about 1 hour and a half now. So I'd like to ask Fred, how much more time do you have for us?
Well, I think I have half an hour.
Yeah, so I may I read some comments here on the room chat. So there's Jada Nation who says that he highly recommends reading the Cyborg Manifesto by Donna Haraway. And there's Ben Hillard who is asking, what about the use of melanin instead of silicon? Can you comment on this? Where are you
Instead of silicon, you said?
Yes.
Mm, but we are not using silicon at this point.
Yeah. So B wants to come up on stage, so I brought him. B?
Yeah, thank you. I just really appreciate this conversation. Fred, I was wondering if you could talk about some of the ethical concerns that have been raised around the biocomputing technologies. It sounded like maybe you've been able to keep some of these organoids alive for multiple years. I think in other media I'd heard it was only about 100 days. So there's like the lifespan concern, but then also just, I guess, the just initial response that many people have, the idea of possibly a sentient organoid being subjected to these demands of computing, and just wondering if you've thought about them. I'm not really taking a strong position myself, but just wondering if you've considered that and meditated on that.
Yeah, I mean, I think it's a very important question, and thanks for asking. So just about the technical question about the duration, there are two different things. 3 months or 100 days is once we put the organoids in connection with the electrodes. Okay, and if we don't put electrodes to them, then we can keep them for years. The problem is that we don't communicate with them. Okay, so now for the ethical aspect. So at this point, we are using a few thousands of neurons. Okay, so at this point, at this very moment, I'm not sure that there is a problem right now, but the problem is more in the future. What about 100 billion neurons that like us, or even more? Okay, there is no limit actually potentially. Okay, and the short answer is that I don't know. I just don't know. And this this is an open discussion. I'd like to open the floor with. We are actually seeking, actually actively right now, discussion with the philosophers or ethicians in universities that could publish about this topic, and we'd be happy to bring the science on our side. But I mean, I would say to some extent we are only scientists. Okay, so I'm a bit out of my field of competence, of expertise here. If when we start thinking about the ethical implication, there are for sure. We have to consider them, and I don't know how to handle this. I don't even know. Should we think about in the long term some regulations like, okay, we can make biocomputers, but they should have less than 10 billion of neurons? um, like a quantitative limitation, or does it make absolutely no sense? I'm really lost on this, I must say.
I appreciate the transparency there. I guess I'm wondering how like distress in an organoid could be measured, right? And maybe it would require studying sort of the baseline behaviors of like what it does when there aren't electrodes attached to it. I don't know how many observations you can share about that. Is it just sitting there absorbing nutrients, or is it having some sort of baseline activities that could be measured and then compared to um, what the behaviors or measurables are when the electrodes are attached, and is there any possible way to, I guess, quantify stress in an organoid?
So there is one way of quantifying organoids. This is, brainstorm. That would be to use some of the metabolites of organoids. That would be one interesting way that we've been discussing actually here also, because the health of the organoids are also important for us. I think that would be the way I would take metabolites, because we have access to the medium, and so we can run analysis on the medium, and in the medium we get a lot of information about the activity of the neurons because they release some molecules. So this would be one smart way to characterize the stress. That would be my guess.
Well, I look forward to following your company and its collaborations with philosophers and ethicists on this question. It's a very important one, and of course, there's so many positive impacts this technology could have in the human world, of course. But being mindful of any suffering that the organoids might experience is I think an important question. So thank you for even thinking about it.
No, you're welcome. And the thing is that I'm very aware also that every time science makes progresses, advances, it gives us new challenges, a new question. And the only thing we can say about advance of science is that if we look back 1,000 years, we live actually better than 1,000 years ago. So the overall benefit is positive. So that's at least one good thing to have in mind. But of course, each time we bring some new solutions with technology, we bring also some new problems. So it's inevitable, I think.
Yeah, Fred, I was also about to ask this question about the ethics because I know that the EU is very strict about the guidelines for this kind of researches. So I think I just read about Horizon 2020 and Horizon Europe, whose frameworks emphasize the responsible use of human biological materials, including organoids. So these guidelines promote respect for human dignity, privacy, and protection of personal data. Although I don't know much about, I haven't read about the details of this. And I think there are scientific societies such as the International Society for Stem Cell Research who provides guidelines for stem cell research, including the use of organoids, which I'd like to also find out later on. And these guidelines, according to what I read, emphasize the importance of ethical considerations and suggest that researchers avoid creating brain organoids with properties that could lead to human like consciousness or significant pain perception. So there is because I think we encountered this. There is another researcher um, in the US. He is a Brazilian researcher, and he is also using organoids to study um, neurodiversities, for example, autism. So he is using this in dishes, and he's trying to study that. And I was reminded of these events that we have, and also this one is not about organoids, but I think we had a guest also here who was working on the lab of Jacob Hanna, who is doing this artificial synthetic embryo models. And yeah, of course, a very controversial thing is that in the future he would like these synthetic embryos to be coming from him, from just like clones of himself. So we discussed a lot about ethical considerations here, and yeah, I'd like to really appreciate that you mentioned that. Of course, you are also still studying it, because basically, when some kind of technology comes, and especially if it comes disruptively, it's there already before we have even prepared for its ethical consideration or its possible impact for us. So yeah, I just wish your lab good luck, and I really appreciate it, because it will be contributing a lot to science.
Yes, yes, you're right on all what you're saying. So I didn't know about this. What we mentioned about the EU and the 2020 concern about and the relation with consciousness is. Problem is the relations with consciousness is very between the number of neurons and consciousness is very obscure actually. Consciousness, even the word is not really very well understood, and so yeah, it's just an open debate. At 10,000 neurons, it's okay, but yeah, I don't know for more. I don't know.
Yes, so they need philosophers and scientists trying to discuss about this. We had so many rooms about consciousness here, also in quantum photonics, from the point of view of a neuroscientist, from also the point of view of ethicists, and yeah, other fields of science. But of course, it is still a controversial issue up to now, and we haven't solved it. Actually, in the same case that, of course, these are not neurons, but the lab of Michael Levine, who also produced these nanobots, it is not neuron based. But the thing is that what is consciousness then in that case? Because it is challenging our understanding, scientific understanding, the established understanding that we have about consciousness, because it does not have neurons. However, it exhibited, the nanobots exhibited behaviors that are really, amazing, considering that it has no neurons yet, and it was cultivated on the lab. And when they developed eyes and also mouth, the lab of Michael Levine tried to Picasso it. They tried to rearrange the eyes, not in the usual areas where it is situated, but it reset into the conventional way that we try to know where the eyes and the mouth are. So in that case, there is a big challenge to our issue about consciousness. And of course, I mentioned a while ago about the issue of pain, maybe that some people are trying to raise. So that's why they wanted to establish something about the organoids. And of course, I mentioned EU because I know that compared to other places, I think EU has a lot of guidelines about different sciences that we have, and recently even about artificial intelligence. So yeah, I think that is something that maybe we should discuss in the future. So, yeah, any more questions from the people here on stage? And if, yeah, Evan?
Oh yeah, I thought of one or research one. The best thing I can think of is continuous processing, operational problems for for upstream perfusion versus, conventional batch processing. I don't know if that's relevant.
What was the question? I'm sorry.
Kevin, can you repeat the question, please?
Oh yeah, sorry. Continuous processing. Bioprocessing. For operational problems remaining with upstream perfusion versus conventional batch processing is what I researched. I don't know if that's relevant though.
So you mean you're comparing continuous processing versus batch processing. So can you elaborate about the difference that you see between continuous processing and batch processing?
Well, I'm not sure exactly. Yeah, perhaps that's not as relevant, but I'm not sure if that ties into contamination risks as well. The contamination risks and development and characterization of time. For because the time required to process, the development and characterization, can be substantial. And I don't know. So maybe those two aren't relevant. So hopefully that's not ongoing.
But indeed, if we talk about the whole process of doing biocomputers effectively, okay, if you talk about duplication of learning, for instance, we have some big challenges here, okay, because you consider that each organoid is different, okay. So batch processing, it's a unitary processing. We cannot apply the same algorithm to train organoid from the other one. They are all different, okay. So this is one of the big challenge. When you have a software, you can just copy it and run it on another computer. It's instant. Here, the hardware and the software are mixed somehow. Okay, so we have to retrain the full wetware each time we want to train something. I guess this is one of the many challenges that we that we have to overcome to have something working working biocomputers. So.
Thanks for clarifying. I'll listen to the recording later. Learn something.
Yeah, thank you, Kevin. So I think we're going to wrap up soon. We've been here for almost 2 hours now, and at this point, I would like to thank Fred for accepting our invitation and for giving a very great presentation and for the very, very interesting discussion. And I could only wish his company, which also has great members, check out their um, profile here on the room chat. You will see the members of his company. And it's a very great research. And we are rooting for you to succeed more and more because we know that this is very relevant to the world. So I'd like to give the mic to you, Fred, now, if you have some messages to us before we close the room.
Well, there is just something that there are two things I wanted to mention on the entrepreneurship and innovation. The first thing is that it has to be a very specific course of event so that we can finance this company. Okay, we had to create a first company from scratch in a garage and be successful and get out all of the shareholders so we can keep all the profits in order to invest in another deep tech company. And I think biocomputing deserves more than only entrepreneurs like us who had this chance. I think funds should invest and venture capitalists should invest into this way more. Only if you consider the patents and the landscape right now, there are so many things to be invented. I think that's just incredible that there are so few companies, and just by chance that we were in this situation. And something else, but still related to money. It's very difficult to keep this track of pure biocomputing. I mean, wanted to compute using neurons. You cannot imagine the number of companies or investors who want us to push into other shorter term application. Okay, and and it's not easy actually when you're at our stage to say no, we want the long-term application, we don't want the short-term application. So these are the two challenges that on the financial and the direction side we have to to control, to make sure we work in the long term on this research on doing a functional biocomputers.
Yes, so thank you so much, and we hope that we can invite you again here in Quantum Photonics next time. How did you enjoy your first Quantum Photonics room, clubhouse room?
Yeah, that was nice. That was unexpected. It was the first experience for me. Um, and what is cool is that it's not stressing at all because you don't see anyone, so it's quite a relaxed conversation. And I was surprised by some people that were saying, "I'm impressed. I'm talking in public," but yeah, yeah. Compared to when you are really physically in front, of many people, that's really a cool way to interact.
Yeah, and probably sometimes you can be a speaker in the room while you are lounging in your sofa and you are on your pajamas. So, yeah, once again, thank you, Fred. We really enjoyed our conversation with you. And I think Bob cannot speak now, so I'd like to thank the people on stage. You have Kevin and B and Xiaoshun, who was here a while ago, and yeah, everybody who came on stage and everyone in the room. I cannot mention all of your names, but thank you, and we will see you in the next rooms. So I'd like to give the mic now to Darcel to help us close the room.
Thank you, Cecile, and thank you, Doctor, for a very great talk.
There's
To close the room,
Darcel, I'm sorry. Before we end, I'd like to say that please try to. I think in a few days this will be in YouTube, so please check it out. So yeah, just search for Quantum Photonics. So thank you, Darcel. Mic to you again.
no problem. So to start our countdown to close the room, we're going to start on lucky number 5. Is everybody ready?
Ready?
Maybe
5.
5.
Kevin 4.
4.
B 3. No B.
3.
Cecile Dos Equis.
Dos Equis.
Good doctor, your numero uno. Call out our last number, please.
The main one.
That's it.
Fred and I will both say number 1. So Fred, let's go. 1.
0.
So thank you so much. Thank you so much, Fred. Thank you so much, everybody. We'll see you in the next rooms.